Lymph is interstitial fluid that gets into lymph vessels. Lymphocytes are T helper suppressor and cytotoxic, B and NK cells. Lymphatic vessels are tubes forming a collection system to return fluid back to the bloodstream. Lymphatic tissue, lymphatic nodules, Peyer's patches, tonsils, and gall. Lymph organs are lymph nodes, the thymus, and the spleen.
Some functions of the lymphatic system include protecting the body by removing foreign material from the lymph in the bloodstream and providing a site for immune surveillance.
Lymph capillaries, also known as terminal lymphatics, are one-way vessels that aren't in bones or teeth. They feature mini valves that let lymph into capillaries, lacteals, which are lymph capillaries in the villi of small intestines, and they absorb dietary fats, and chyle is a form of lymph in the small intestines from dietary fat. The location of all of these features are in the villi of the small intestine. Lymph collecting vessels are a system of vessels like veins that are in all tissues and have more valves and anastomoses than lymph capillaries. The location is scattered throughout the body, following veins and arteries. Lymphatic trunks collect lymph from the lymphatic collecting vessels. There are two ducts. The first one is the right lymphatic duct, and it drains the right upper part of the body, including the right arm, right side of the head, and right thoracic region. This duct empties into blood vessels between the right internal jugular and subclavian vein. The second duct is the thoracic duct, the bigger of the two ducts. It drains everything else and empties into the left internal jugular and left subclavian vein. The cisterna chyli collects lymph from the legs and abdominal area. Lymph transport. Mechanisms for pumping lymph towards the heart. The respiratory pump moves the lymph through the abdomen, where pressure increases above the diaphragm and decreases below it. Lymphoid cells. Lymphocytes are produced in an epiphysis of long bones, the red bone marrow. Regulatory T cells control immune response. The helper T cells supply stimulus to begin an immune response. The suppressor T cells slow down or stop immune response. The cytotoxic T cells target and destroy infected body cells such as viruses or cancer. B cells form plasma cells to make antibodies and need a stimulus. The NK, natural killer cells, which are immune surveillance, attack virus and cancer-infected cells and don't need stimulus. Macrophages eat bacteria, invaders, and cellular debris. Dendritic cells are spiky and found in lymphoid tissue. Reticular cells make reticular fibers, and reticular fibers provide places for cells to hang out.
This is going to be a very brief overview about humoral immunity and cell-mediated immunity. In both, we start up here with the pathogens. So these sausage-like figures are known as the bacteria, and the little stars are the viruses. So first, the macrophage will phagocytize the bacteria. Once it becomes inside, it will chop it up into little pieces in this circle right there. After it chops up the bacteria, it becomes MHC2, which is major histocompatibility complex 2. And this is only in immune system cells, and this is known as holding pieces of bacteria. Here, on the macrophage, the pieces of bacteria are on the outside of the cell. The macrophages are showing that what they have outside of them. These are called antigen-presenting cells, abbreviated here as APC. So by definition, APC is a macrophage that has caught a bacteria and says, look, here's the bacteria. So in this picture, this is the MHC2, the structure right there, and the whole thing as a total is the antigen-presenting cell. And these orange things are the bacteria chopped up and on the outside of the APC. So after we form the antigen-presenting cell, we're going to move up here. So we need to get a helper T cell involved. Helper T cells are going to help the immune system. Without helper T cells, we wouldn't have an immune response. When a helper T cell is just sitting right here, it is naive. That means it has not been activated yet. The APC wants to show the helper T cell what it has. Likewise, the helper T cell also wants to see what the APC has. Now that they have shown each other what the APC has, it is not naive anymore. Now the helper T cell is going to divide and differentiate into active T cells and memory helper T cells. By the way, where this is happening is in lymph nodes. It can also happen in different parts of the body, but we're going to just focus this on in lymph nodes. So hanging out in the lymph node is also B cells. These are also naive, meaning that they haven't been activated yet. It also holds antibodies on the outside. Okay, along comes the bacteria. And the bacteria sticks to these antibodies. The B cell now notices that he's caught something, so now it no longer is naive. Once the B cell knows he caught something, now we're going to need help from the helper T cells to activate this. So the helper T cell is going to send the active helper T cells. So here comes the active helper T cell is going to talk to the B cell, and it's going to provide a chemical which is called interleukin-2, also abbreviated as IL-2. Interleukin-2 is going to activate the B cell. Once the B cell is activated, it's going to divide and differentiate into memory B cells and plasma cells. Plasma cells will divide and differentiate to produce boatloads of antibodies, as seen right here. And this altogether, this side, is called humoral immunity. Now, let's say we have a virus, and the viruses are depicted as little stars. The MHC on the outside of the cell, and this is simple columnar epithelium in the GI tract. MHC1, which is depicted as this right here, is on every body cell except red blood cells. MHC have normal cellular proteins. If a virus gets into those cells and starts reproducing, the cell will say, uh-oh, I have something bad inside of me. And it's going to push it out into the MHC1, like it is right there. Now, after pushing it from the inside out into the MHC1, MHC will say that it needs help. Now that it is on the outside, we're going to need a cytotoxic T cell because those are the cells that kill. So the APC, the antigen presenting cell, will bring over a helper T cell. The helper T cell will give interleukin-2, abbreviated as IL-2, to the cytotoxic T cell. Right now, the cytotoxic T cell by itself, without the helper T cell and the interleukin-2, is naive. This means it hasn't been activated yet. Once activated by IL-2 from the helper T cell, it will divide and differentiate into active cytotoxic T cells and memory cytotoxic T cells. Once it becomes these two cells, the active cytotoxic T cells will go in and kill the viruses on the outside of the MHC1. This is all known as cell-mediated immunity. Lymphopoiesis is the formation of a lymphocyte. A hemocytoblast in the bone marrow is an immature lymphocyte that is released. It then goes to the thymus, which is T-cell differentiation, also T-cell maturation. It's like the T-cell ninja school. This then becomes a T-cell, and this happens in the bone marrow, spleen, and other peripheral tissues. It also can go into the bone marrow. This is where it becomes a B cell. This is in the lymph nodes, spleen, and other tissues. It can also become an NK cell, which moves out into the tissues. Lymphoid tissues house lymphocytes and immune system components. Their functions are immune surveillance, watching for bad invaders. Their structure is reticular connective tissue, and this makes up most of the lymphoid tissue. Types of lymphoid tissue. Diffuse lymphatic tissue is found throughout the body in chunks. Lymphatic follicles, also known as nodules, are lumps of lymphatic tissue that contain germinal centers. Germinal centers are light areas surrounded by dark. They divide and become plasma cells that make antibodies. Tonsils are the simplest lymphoid organs. They form a ring of lymphatic tissue around the entrance to the pharynx, where they appear as swellings of the mucosa. They contain follicles with germinal centers, and they are surrounded by lymphocytes. The The Palatine Tonsils are located on either side of the posterior end of the oral cavity. They're the largest tonsils and the ones most often infected. The Pharyngeal Tonsils, also known as your adenoids, are located on the posterior wall of the nasal pharynx. The Lingual Tonsils are a lumpy collection of lymphoid follicles at the base of the tongue. Their functions are to remove The functions of the tonsils are to remove pathogens from air and food, keep the immune system activated, and trap bacteria. Tonsil structure. There's nodules containing germinal centers and crypts, which are cracks in the tonsils that trap bacteria. Mucosa-associated lymphatic tissue, also known as MALT, equals gut-associated lymphatic tissue, which is GALT, and GALT is aggregation of lymph nodules. MALT and GALT locations. First is Peyer's patches. They're found in the small intestine and their function is to destroy bacteria. Second is appendix. Appendix is a long tube that hangs off the large intestine and it's filled with nodules. 3 is bronchi, and bronchi are the passageway to the lungs.
MALT and GALT functions. They trap pathogens, engage immune system.
Lymphoid organs, lymph nodes. Functions of the lymph nodes include filters lymph fluid, removes bacteria and viruses, and also participates in immune system activation. Lymphoid organs, lymph nodes. 2. Structure. Capsule with trabeculae. Walls off of lymph node from the rest of the body. The trabeculae divides it up. Stroma is the framework of the reticular fibers, and cortex is the outer part of the lymph node. The medulla is the middle part of the lymph node. It has macrophages. The medullary cords are pathways of the same tissues that makes up the cortex. It contains B and T cells. Lymph sinuses are where you find macrophages, passageway through the tissues. Circulation in the lymph nodes. Afferent lymphatic vessels are going towards the lymph nodes and bringing fluid into the lymph nodes. Efferent lymphatic vessels carry lymph fluid away. Location is clustered along lymph vessels. Concentration in cervical, axillary, and inguinal.
Thymus gland. Location. Lower part of the neck under the sternum. Most active in children. Functions. Site for T cell maturation. Produces hormone thymosin, which causes maturation. Spleen. Functions. Site for making lots of lymphocytes. Cleans the blood. Stores iron for making hemoglobin. Makes red blood cells in fetus. Stores platelets. Hilum, where splenic artery and splenic veins meet. White pulp, cluster of lymphocytes around the arteries in the spleen for immune surveillance. Red pulp, everything else. Red blood cells recycling, cleaning the blood. Clinical aspects. Lymphedema, swelling by blockage or removal of lymph vessels. Buboes, swollen infected lymph node. Splenectomy, surgical removal of the spleen.